is that it's not always easy to define a single endpoint to evaluate a treatment. So how can we combine them? That's the topic of today. And very specifically, how can we combine them also with patient-relevant endpoints, such as quality of life, which is indicated as well very nicely from the respondents, that it is also interesting endpoint to include in the effective evaluation of novel treatments. So this is the outline of what I would like to go over with you today. So first we will talk about ways to combine multiple endpoints in clinical trials. And very specifically, we will look into the ability to combine data types of different data. So that means that we want to include, for example, a quality of life, which has a different data structure than, for example, a survival outcome. So how can we do that to combine them? And also, of course, since this is a rare disease audience, we'd also look at the abilities of these methodologies to cope with very small samples. One of those methodologies I will pick out. I might be slightly biased here because this is one of the major topics of my research, but I do believe that the general pairwise comparison methodology is very useful to combine multiple endpoints, including of different data types, and also applicable in small samples. So we go over that into detail, and then finally I will illustrate the theoretical development, if you will, which I try to keep as little as possible, to a rare disease, a skin rare disease called epidermolysis bullosa. So I've had the pleasure to be involved as well in an EJP RD projects under the guidance of Johan Bauer, also present here today, and Jörg Zimmermann from Austria. So what is actually recommended by the International Conference Council of Harmonization is that if you want to run a clinical trial, then it's best to find a single meaningful endpoint. Now, as very nicely illustrated by you, in fact, is that that's not always easy to define. And that may be for various reasons. One of those reasons may be that the disease itself has a multifaceted outcome. So there is not a single outcome that might reflect the full clinical benefit of a treatment. So it's very difficult to find just a single endpoint to see, okay, well, if this changes, then we know that our treatment works. So the solution is then, well, we need to combine these endpoints, clinical meaningful endpoints, but in which way can we do this? And very specifically, as I said, we will look at a few possibilities, and we will specifically look at, can we combine data of different type so that we include quality of life, which you indicated is also important, and what are their properties for small samples? Can we use them as well in rare diseases? So first of all, how can we combine the endpoints? Well, we can combine them on a subject level. So every subject in a clinical trial thankfully gives us data that we can use. And now we can try to combine all of that information on a subject level, which is in fact a quite common methodology, not only in rare diseases, but also in large studies such as cardiology and oncology. And what we try to do is we try to reduce the multivariate outcome to a single univariate endpoint. For example, we can use clinical indices. Clinical indices are, for example, a lot used in clinics. Physicians, they know these very well. So what we do is we look at certain lab values or radiographical examinations. We score them, and then we sum some score, and then we have a clinical index that gives you an idea about the health status of a patient. So that's one way to do this. Another one, which is quite often used in again, cardiology and oncology, is what we call composite endpoints, is where we have multiple time to events. survival data, which, for example, mortality, myocardial infarction, hospitalization, those are all survival data, time to a certain event. And what we do is we try to reduce those multivariate endpoints to a univariate endpoint by taking only the first event that happens in the patients. And there are quite a lot of methodologies that are quite standard in statistics. So we have the Cox proportional hazard model. We have the log rank test, not the long rank test. I'm sorry for that. That's the log rank test, of course. And we can extend them as well, because it has realized that, okay, we have multivariate outcomes, but we reduce it to a univariate endpoint, which is not ideal, of course, because very often, for example, in cardiology, when we have a multivariate outcome, we have mortality, myocardial infarction, hospitalizations. Very often, of course, hospitalizations are much more frequent, luckily, than mortality. But also they are usually earlier in time than mortality. So that means that if you reduce from a multivariate to a univariate outcome, that you are actually evaluating mostly the clinically least interesting endpoint, which is the hospitalization. So that's why people have tried to extend these analyses to include also the other events that the patient has. So there's Anderson-Gale, there's Wiley and Weisfeld. There's a lot of extensions of the Cox proportional hazard. And recently, there's also been an extension of the log rank test, which is called the weighted composite endpoint, where we try to give a weight to each of those events. So normally the log rank gives a weight of 1 to the first event that the patient has and 0 to the subsequential events. Now, what they do in the weighted composite endpoint is they give a weight between 0 and 1 to each of those events. Now, the downside being of that is that each weight is counting much less than 1. And in fact, we've seen that the power of this test is quite low. So it may not be a very interesting way to go forward. This is one way to combine multivariate outcomes on a subject level. Another one is, for example, we try to use models, semi-parametric or parametric models, where we try to model each of those outcomes in a joint model, a joint frailty model, for example. There are multiple models, but usually these models are restricted. They are restricted in the number of endpoints that you can combine. Also, the type of data that you can combine is restricted. For example, the composite endpoints, where we look at the time to first event, we can, of course, only look at survival data. We cannot look at quality of life together with survival data. That's not possible. Often these methodologies also have a very poor small sample properties. And in fact, you showed indeed that these parametric and semi-parametric models, so here and the composite endpoints jointly together is indeed quite often used and one of the methodologies that you have indicated to prefer, next to also the multiplicity correction. I will come to that in just a second. So this is one way we can combine at the subject level. Another way is we can try to combine at the test statistical level. So what that means is that we try to compute a statistic from the data for each of those endpoints, and then we try to combine them in some way. And there are a few methodologies, not much. One of them is suggested quite some time ago by O'Brien, which combines t-statistics. But t-statistics, they can be used only on continuous endpoints. So again, we have an issue here if you want to include quality of life, which typically is not continuous. The benefit of the methodology is that it accounts for correlation. And that is actually a very important point, because once you have multivariate outcomes, there's also going to be some level of association between those two endpoints. And it's very important to capture that as well, as specifically if you want to look at, for example, benefit and risk endpoints in a single analysis. So it's very important to be able to establish the association or take account of the association between these two in a single analysis. And another methodology where we combine test statistics or multivariate outcomes on a test statistical level is called the average Z-score. So there again, we calculate Z statistics and we take the average. The benefit of this is that it's not limited to continuous endpoints, so we can combine any type of outcome that we want, but it does not take account of the correlation. So again, it has some limitations. And finally, and that's the other option that you've indicated, we can try to play around with the p-values to combine at the p-value level. So for each of these outcomes, we are going to use a test statistic that you can use for that specific outcome. And then we're going to try to combine those p-values at different levels. So there are a few procedures. Some of them can actually take account of correlation, but most don't. And then, yes, the multi-testing procedure, in fact, account the multiplicity correction as one of those methodologies. But as statisticians would tell you, multiplicity corrections, when you have multiple outcomes, your power is reduced. So it may not be the ideal way to go forward. So in fact, when we look at the whole literature and classes of ways to combine multivariate outcomes, each of them has at least one or a few limitations. Either they ignore the correlation between multivariate endpoints, they're limited to a certain type of endpoints, only survival data, for example. They treat the endpoints as equally important, which is very important to take into account. For example, if you look at mortality, myocardial infarction, and hospitalizations, there's a clear clinical importance in these endpoints. And what we do with all these methodologies that we just discussed is that they treat each of these endpoints as equally important. But perhaps a clinician, a patient, would like to give more importance to one of those endpoints. For example, as a patient or a physician, you do not want your patient to die. So that may be much more important than just a minor hospitalization, for example. So these methodologies that we just discussed, they cannot take that into account. Also very important, what we want to do evaluating a new treatment, we want to know whether the treatment works, yes or no, which is called the hypothesis testing. Once we know that, we might also want to get an estimate of now how much does it work. So that's what we call an effect size measure. And most of these methodologies that we just discussed, they do not have a very clear effect size measure. So we can only say, well, yes, it works or it doesn't work, but we cannot say how much it works. And so we cannot compare one treatment to another, for example, from two distinct clinical trials. So that is also something that is very important that ideally we would need. And then finally, most of these methodologies, they have very poor small sample properties. And so indeed, these limitations that I just named, they come back again in your answers to the questionnaires. Indeed, indeed, a majority of you has difficulties with the number of outcomes, the type of endpoints that can be combined of the both together. And indeed, as we have an audience here for rare diseases, the poor sample properties of these methodologies is also quite a big limitation. Now, can we do better? Well, very recently, the last decade, there are a number of non-parametric methodologies that have been developed to combine multivariate outcomes. So one class is based on ranks, the global rank of the desirability of outcome ranking. So what you want to do is just, you can rank the outcomes. For example, mortality is ranked first, and then, for example, myocardial infarction, and then hospitalization. And what you do is try to rank the outcomes of the patients on mortality first. If there is a tie, then you try to rank them on the next outcome. And you go forward, and then you can construct statistics on that. Now, this is a very elegant way, but the problem with the ranks is that once you have sensor data, which happens in survival data, then it's no longer possible to have an unambiguous rank, meaning that if you have a patient A which is doing better than patient B, a patient B doing better than patient C, then you can end up in a situation that patient A is actually doing better as well than patient C, instead of what you would expect, the reverse. So that is a problem with ranks in multivariate censored outcomes, which you do not have with something that we will discuss in detail now, which is the generalized pairwise comparisons, which are an extension of a Mann-Whitney test. But wait a minute, statisticians will say, well, the Mann-Whitney test is also a rank test. Yes, that's true. But equally, you can use pairwise comparisons to come up with the exactly same the Mann-Whitney test. And we will see next that this can be extended to multivariate outcomes. And I'm very happy also to have Mark Buys as one of the panelists here, because he's one of the, I would say, the founding fathers of general pairwise comparison. In fact, he's the one who suggested the name of this methodology. So I'm very happy to hear his comments later after the presentation. In the literature, you can also find them as win statistics. So Kaung Dong is also very active in research in general pairwise comparison, but he in fact calls it win statistics. So now, what is this general pairwise comparison methodology? Well, as I said, it's actually an extension of the Mann-Whitney test, and Mann-Whitney test is just for univariate outcomes with uncensored data. Which has been quite a long time ago also extended to censored data, so for survival data, which is called then the Guillen and Gilbert test. Actually, Guillen and Gilbert independently suggested the same test, and which is now extended to multivariate censored and uncensored outcomes, and we call that general pairwise comparison methodologies. Now, the most used generalized pairwise comparison methodology is actually the one that prioritizes first the outcomes. So we call it also prioritized GPC. So you rank first the outcomes that you have by usually severity. So the most severe outcome is ranked first. If you're in a cardiology trial, that is usually mortality. What you then do is you construct all possible pairs that you can construct between two subjects, one from each of the treatment arms. And per pair, you're going to look which of those subjects is now actually doing better on that first ranked outcome. If the group X here on the slide is your new treatment that you want to look, if that patient is doing better, then that pair gets a score of 1. If the patient X is doing worse than the patient in group Y, the comparator, then it receives a score of -1. Then you were able to score the pair and the algorithm stops there and you go to the next pair. Now, if there's a tie, for example, for mortality, none of the two patients died, then we have to go to the next outcome, next ranked outcome. For example, myocardial infarction, and we do exactly the same thing. See if we can decide which of those two patients is doing better. If we can, it stops there. We go to the next pair. If we can't, we go on and on until our last outcome. And if we then cannot decide which of the two patients is doing best, then we have a tie for that pair. Now, in the end, what we get is actually a count of the number of wins for our new treatment and a count of the number of wins for the comparator. And with those, we can construct statistics such as the net benefit or the net treatment benefit, I should say. And if you take that difference and divide it by the amount of pairs that you have, we actually have a statistic that has values between -1 and 1. And that is very convenient because that means that if the result of your analysis, the net benefit gives you a value above 0, that means you're better off with a new treatment than the comparator. If that value is below 0, then it's actually not a good idea because then it's better the comparator than the new treatment. And so we can translate the net benefit also in probability. So it's in fact, it estimates the probability that a patient in the new treatment will do better than a random patient in the comparator minus the reverse. Now, very conveniently, it's also a U statistic. So there's a whole theory on U statistic that you can use also on these net treatment benefits. And lastly, it's also related to something that is called a probabilistic index, the relative effect, which is around for quite a lot of time as well. Also in ROC analyses, in stress strength analyses. So it has a very natural connection as well with another large part of the literature. Now, this is an absolute treatment effect that we can define, so a difference between probabilities. But we can also define a relative treatment effect, the win ratio. So we just divide the number of wins in the active treatment arm and the number of wins in the comparator, which has then as values between 0 and infinity. And if the value is above 1, that is in favor of the new treatment. If it's below 0, then it's in favor of the comparator. But it has been criticized, in fact, that it ignores the number of ties that you have in the comparisons. And so only very recently, that has been extended to the win-odds ratio, which includes now the number of ties. Again, it's a relative effect measure, so it has values between 0 and infinity. And very conveniently, it's just a transformation of the net treatment benefits. So in fact, you can just do a statistical test on the net treatment benefit, for example, and then you can express the extent of the treatment effect in a absolute treatment effect, the net treatment benefit, or in a relative treatment effect, the win-odds ratio, which is in fact exactly the same since it's a transformation. Now, I've talked about the prioritized general pairwise comparison, where we prioritize the outcomes, but there is also a variant that has actually, in not exactly the same way defined, but related by O'Brien again. So besides the semi-parametric and parametric t-statistic combination of multivariate outcomes, he also suggests a non-parametric combination, and actually you can extend that and that's what we call now a non-prioritized general pairwise comparison method. So in fact, instead of prioritizing the outcomes, the multivariate outcomes, you now do a pairwise comparison for each of the pairs on each of the outcomes that you have. And so again, that leads you to a number of wins in the treatment arm and a number of wins in the control arm, which, for example, we can still use the net benefit as statistic, but now we also divide by the number of outcomes that we have. And that makes sure that we also, we again have a statistic that has values between -1 and 1, and that's easy interpretable. So in fact, general pairwise comparison is a framework that is quite flexible. So we can decide, and the decision perhaps should be made on clinical motivations. So if there's a clear prioritization in the endpoints that you want to evaluate, and a prioritized GPC may be of interest. If it's not possible to give a clear prioritization to your endpoints, then you can also use a non-prioritized analysis. We can also match pairwise comparison. And what do I mean with that? It will come back later as well. You can compare each patient of each treatment arm with each other, or you can compare just one patient from one treatment arm to one patient of the other treatment arm, and not the rest of the patients. And that is interesting for matched designs, pair designs trials. For example, in eye studies, one eye is treated in a patient, another eye is not treated. It's the same patient. So to take account of the within correlation, you can use a matched pairwise comparison where you only compare the outcome of one eye in a patient with one eye of the other patient, and you do not compare to the eyes of the other patients in the study. And finally, as I said, we need to define which of the two patients is doing better. Now, if you have a continuous outcome or a survival outcome, for example, you can say, well, that patient died on day X and the other patient died on the day after. That's the difference. So the second patient is doing better. Or you could say, well, a difference of 1 day is not really a difference, clinically speaking. So we just extend that and say, for example, well, the difference between those two patients is only a difference if they die a week apart from each other. So that is the threshold of clinical relevance. So that's also a possibility to include in your analysis. Now, of course, these things you need to set pre-analysis of your data. Now, GPC is quite new, so what we did the last few years, and I will not go too much into detail, is to look at some of the characteristics of general pairwise comparison. And what we know, if we have a univariate outcome that's uncensored, we know it's unbiased and very efficient in clinical trials scenarios. When it's censored, there's a bias, but it can be corrected. And then importantly as well, the correlation in a multivariate outcome is captured by these prioritized and non-prioritized GPC, but in a different way. So the prioritized GPC, when we rank our outcomes, then your statistic itself, so the net benefit, for example, will change depending on the correlation. So you will see on the effect size measure what in fact, or it will change in fact with the correlation, which is interesting, for example, in a benefit risk analysis, for example. And then in the non-prioritized GPC, the correlation is not captured in your effect size. So that will be exactly the same, irrespective of the correlation, but it will be captured in the variance of the test statistic. So in fact, the hypothesis test of both the prioritized and the non-prioritized GPC will take account of the correlation between the multivariate outcomes. Now, inference, that's what we need for hypothesis testing, and that's what we are getting slowly towards rare diseases. It's all nice and well, but does this work in small samples? As I said, it's a topic, GPC, that has been the last decade received a lot of attention in research, and so there's a lot of tests, hypothesis tests that has been suggested, such as a resample permutation test, resample bootstrap test, rank-based test, and also use statistic tests. But for the statisticians, you see asymptotic use statistics. So asymptotic means that you actually need a large sample size for the hypothesis test to be valid. So is that useful in small samples, question mark. Now, also very conveniently, I've shown you here the tests that have been suggested for net benefit, the win ratio, and the win odds. But in fact, we can show theoretically that all these tests will lead to exactly or approximately the same p-value. So whether you use the net benefit, the win ratio, or the win odds in your hypothesis test, that doesn't matter because they will give you approximately the same p-value, which is good news. But now, small sample behavior. Well, in fact, if we look at the Guillen test, the Guillen and the Gilbert test, remember that's the univariate extension of the Mann-Whitney to sensor data. And they, quite some time ago, they suggested a test, which we call now the exact permutation test. And what that does is, in fact, we are going to permute the data that we have. And if we take every possible permutation of the data, calculate there the GPC statistic, the net benefit, for example. We have a bunch of net benefits. Then we can make a distribution of that. Well, that distribution under the null hypothesis is standard normally distributed. And they showed already back in the 60s of previous century that this exact permutation test works for very small sample sizes, even up to 5 patients in each treatment arm. And indeed, we, when applying these exact permutation tests on the general pairwise comparison, you can see on the top row, which is the net benefit, and the bottom row is the win ratio, or in fact, the log of the win ratio. And you can see on the left column that indeed these permuted data is normally distributed. And also on the right, you can see the normal QQ plot, which shows that even for very small sample sizes, 5 subjects per arm, so a clinical trial of 10 patients only, the normality assumption is is valid, and so the test is valid in very small sample sizes. We did also a large simulation study where we compared all those different tests that exist. So you see the first three columns, that's the U statistic. So that's the one that actually needs a larger sample size. And you're looking at the type 1 error for the non-statistician. So we want to see here a value close to 0.05, very close to 0.05. So you see that those U statistics, they only start to work when you have a study of at least 100 or 200 subjects, while the exact permutation, indeed, for very small sample sizes, it controls the type 1 error very nicely. We extended the exact permutation also to an exact bootstrap, but that doesn't seem to have the same quality of small sample properties than the exact permutation tests. So in fact, those GPC, they can circumvent all of the limitations that we've just mentioned previously on multivariate outcome combination. So it does capture correlation between the endpoints. You can use any type and any number of endpoints and combine them. You can even decide on a priority of your endpoints so that you give more priority to the more clinically severe endpoints. It has a very straightforward effect size measure, well, even several. You can choose between an absolute and a relative effect size measure, and it has very good small sample properties. Now, all nice and well, you would say, this is theory, but now what about practice? Well, in fact, back in 2019 and 2020, not so long ago, a drug called Vyndaqel was approved by FDA and EMA based on an ATTRACT trial, and that ATTRACT trial, in fact, had a primary endpoint based on a prioritized GPC. So it was based on mortality and hospitalizations, prioritized in that order. And for a disease, and so the drug was approved for a disease called amyloid cardiomyopathy, which had an orphan drug status. Well, you can question that perhaps if you can do a study with 441 patients. I think some of the listeners would dream of being able to do a study that large. But in fact, so both the FDA, the EMA, regulatory authorities have already been exposed to this methodology and have approved the drug for a rare disease indication. Now, the topic of today is also how can we combine endpoints with quality of life? Well, I can tell you as well that there is a study in cardiology running currently that has a primary endpoint or a primary analysis also based on a prioritized GPC that includes a quality of life. So more and more these methodologies being picked up, and indeed regulatory authorities are open for this novel methodology. Now more practical. As I said, I've had the pleasure to be involved in the EB-StatsMax project, which is part of the EJP RD program in the demonstration project of existing statistical methodologies, where we work together with well, actually a large group from Salzburg, from Austria, and a group in Sweden, Uppsala, together with the Hasselt University, and so it is a project around epidermolysis bullosa, so a rare skin disease. So in fact, it's mostly based on a study published a few years ago in Wally et al., a single trial that had a crossover, a longitudinal design. So epidermolysis bullosa patients, it's a rare skin disease, so they have a genetic disorder where the part of the cytoskeleton of the skin disease is malfolded. And so with the least mechanical stress, those patients develop painful blisters. So you can imagine that also the quality of life of these patients is quite heavily damaged, if you will. And so this trial looked at only 15 pediatric patients, so a very small sample size, where patients were treated every day during 4 weeks, so almost a month, and then followed up for 3 months. And after that treatment, there was a washout period, and then patients were crossed over to the other treatment. Now, the primary endpoint was they looked at whether a patient had a 40% reduction in the number of blisters that that patient developed on a certain area on the skin at week 4. Because they used, in fact, a Barnard test, which for the statisticians, it's a bit similar to a Fisher exact test, but slightly better properties. And in fact, the Barnard test, it's based on a 2 times 2 table. So that means that you can only look at one time point at week 4, and you have to look at the 2 treatment periods separately. And in fact, the conclusions were that on the first treatment period, there was a clear benefit of the treatment. In the second period, not so much. So this is, from a statistical point, a bit awkward to give a definite conclusion. So ideally would be that we would be able to combine everything in a single analysis that would show you a treatment benefit. And so that is indeed what the aim is of the project that I was involved in. So how can we use the data of this trial in a better way? How can we use all of the longitudinal data? So the blister count were in fact measured at baseline, at 2 weeks, at 4 weeks, and at 3 months. So how can we use all of that information? And secondly, specifically for this audience, there was also a quality of life measured for these patients at baseline and at post-treatment visit at 4 weeks. So how can we combine that with the blister count information? Well, to be honest, the project itself is mostly based on the longitudinal data, but out of interest, I included as well the quality of life. It's a bit of a side project, if you will. And so within that multidisciplinary group, we actually look at different types of tests to look at the blister outcome. So we have a few non-parametric tests, a rank-based marginal model, GPC, of course, since I was involved, a semi-parametric model, general estimation type of model, where we can use small sample corrections, and also model averaging in parametric models. And so we did a lot of simulations with these methodologies on the EB trial. And what we've seen, in fact, is that if you look at the power, so the power to detect a treatment difference, it's best or the highest in those GE type models. And these are just names for small sample corrections. And so important is, well, one, you have to look at the type 1 error control, certainly in small sample sizes, which is controlled in this case. And you compare then for the ones that control the type 1 error, you just control the power. And of course, you want to have the test with the highest power. Now, if you look at the Barnard test, the test that has been used in the primary analysis, you see that it doesn't even really control the type 1 error well in this small sample particularly. Also, the other non-parametric methods, including the GPCs, in fact, they do control, well, except for the match, but I will just discuss this later. The unmatched GPC, if you will, they control the type 1 error well, but the power is not as much as the GE type models. And in fact, it also has a quite straightforward interpretation. So you can use an odds ratio, and then in fact it uses the longitudinal data, the crossover information, and you have a single analysis with a single p-value, which shows that indeed Diacerin, so the cream that's applied to the skin in this treatment compared to placebo, is indeed much better than placebo, and the treatment effect has an odds ratio of 5.7, and it has a quite large confidence interval, but that's not unusual, of course, with a very small sample. Now, also interesting is indeed that we can look at the difference between the first period and the second period within the same model. And there we see as well that what we've seen before with the Barnard test, that the first period, the treatment effect seems to be much larger than the second period. And in fact, within the same model, you can still look at a carryover effect. And it showed that, in fact, the washout period might not have been long enough in this trial, because there was an indication that there has been a carryover effect in this trial. So the fact that the second period does not show a very large difference may be due because of the carryover effect that played there. Now, I still didn't talk about quality of life, which is one of the biggest part in the title here, and you've indicated that it's only 2% of you said that's never important, but most of you agree that quality of life is indeed important to evaluate. And I would argument that in the case of epidermolysis bullosa, it's indeed important to look at quality of life, because the hindrance in those patients in their daily activities is quite high. So we're looking at the data of the EB trial that I introduced. They used a quality of life questionnaire based on 8 questions, which each were scored between 0 and 3, depending on the hindrance that they had at their daily activities. And so the maximum score would be 24, and the lower that score, the better the quality of life of the patient, of course. And then looking at the 2 treatment arms, if you take all of the treatment periods together, then there seems to be quite an indication that the Diacerin treatment, which does not only decrease the blisters, as we've just seen, but also seems to decrease or improve the quality of life. We can also look at in the bottom figure at the 2 treatment difference, the 2 treatment periods, excuse me. And there again, we see that perhaps in the first treatment period, there seems to be a bigger effect than in the latter. Again, possibly due to the carryover effect. Now, importantly, previously we discussed longitudinal outcomes. Now, the quality of life is only measured at the post-treatment visit, so week 4. So now we cannot use a longitudinal information. We just look at a single time point, just to be clear about that. And then looking at all of the methodologies that we looked into in the previous exercise, actually only general pairwise comparison methodology can include quality of life in the analysis. And there's different ways how we can do that. So there's the prioritized general pairwise comparison, where we first rank the clinical outcome, the blister reduction, and secondly, the quality of life. So only in those pairs where there is no difference between the blister reduction, then we go and look to the quality of life. We can do a non-prioritized, where we evaluate all of the pairs in each of the 2 outcomes. And then there's something which is called the matched prioritized GPC, but because in fact we have a crossover trial, a single patient is treated with both treatments. So instead of comparing each of the subjects with all of the other subjects, we might want to look as well to a single patient and compare their outcomes only within the same subject. So that is the matched prioritized GPC. Now we need as well there a different way of inference for this matched GPC. And if you look into the literature, in fact, the uniformly most powerful test for a matched analysis would be a conditional sign test. But unfortunately, the references in the bottom, they looked at a simulation study, and they said, well, you would need at least 15 to 20 patients, paired subjects, for these tests to be valid. In fact, it also ignores the number of ties, and Konieczny and Pauli actually motivate that under certain conditions, and they're applicable to the exact permutation tests, you can just ignore the pair design and your test is still valid. So in fact, we may not need this matched GPC in the end at all. So here as well, I did a simulation of the EB trial, blister counts, and the quality of life. What we did is just we shuffled around the data that we have multiple times, and that makes sure that whatever treatment effect that there is in the data disappears. Then we add a treatment effect for both the blister count outcome and the quality of life. And what I did as well is I wanted to look as well to two different ways to look at the blister outcome, which was the dichotomized outcome as the primary analysis, which is a 40% reduction at week 4, yes or no. But that would mean in a pairwise comparison that you would have a lot of yeses and nos, and so a lot of ties as well if you pairwise compares patients. So you lose, in fact, quite some granularity in the data when dichotomizing the data. We can look also at the count itself, but then we need to standardize that. So what we do is we take the count at week 4, subtract that from the count at baseline and divide it by the count of baseline, and then compare those numbers within the pairs. The results, I put a number of patients that we had. I said at the beginning we had 15 subjects in the trial, but in fact, some of the patients are not matched because they left the study and a new patient came in. And so there's in fact only 13 patients that we can match for the dichotomized outcome. So the top of the table here, the bottom part of the table is the standardized difference, and there's only 12 patients because one subject in fact had 0 blister counts at baseline, and since we cannot divide by 0, we need to exclude that patient if we look at the standardized difference. And so what we look at again is this type 1 error that needs to be at 0.05, and then if we look at the matched prioritized outcome, then you see that for the two-sided test, so we can test one-sided or two-sided, you see that it's not even close to 0.05. Here it's slightly better, but still not good. So this matched GPC, or at least our simulation, seems to confirm indeed that below sample size of 15 to 20, that the conditional sign test does not control the type 1 error very well. If we have the unmatched GPC, there we see that the type 1 error is much better controlled, certainly for the prioritized and the non-prioritized. And that indeed, as one would expect, if you simulate a treatment effect on 2 of the outcomes, that if you compare the power of a single outcome on the blister, that the power increases if you add the quality of life. So both the prioritized and the non-prioritized GPC, the power goes from almost 60 to almost 100 when you add quality of life. Now it is slightly less so for the standardized outcome. And that is a very logical explanation, and it makes you think about the design of your trial. As I said, if we take a dichotomized outcome, we have a lot of tied pairs because there's either a 1 or a 0, so there's a lot of tied pairs. And then we go to the next outcome, which is the quality of life, which has a quite large treatment effect. So in fact, the difference between these two is going to be large. Now, if you look at the standardized count outcome itself, we have much less ties because we look at the count itself that we compare. And so it has much more granularity. And so it goes in less pairs to the level of the quality of life. So it in fact uses much less information from the quality of life. And that's why the increase in power when using the standardized difference is much lower than when you use the dichotomized blister outcome. Now, what if we apply this now to the trial itself, the EB trial itself, and we reanalyze the data? Then, as you can see, and we've shown that in the simulations, that the power for a univariate analysis of GPC is quite low. So if you look at the binary count outcome and the standardized difference outcome, then you see the p-values, the 1- and 2-sided p-values. There's little evidence for a treatment effect. But then if we add quality of life to that in a prioritized way or in a non-prioritized way, then you see that in fact all of these tests are showing a treatment effect of indicative of a treatment effect. Now I want to point out as well to the advantage of using a GPC and presenting a GPC in this way. So you can actually, if we just focus on the unmatched prioritized GPC, what you see is you have the rank of your outcomes, and you can actually look at the net benefit at each level of those outcomes, and actually see what the contribution is of each of those outcomes to the overall effect. So that is an additional benefit compared to other methodologies, which are sometimes more of a black box. There's one number coming out, and you don't know where that number comes from. Is that from the binary outcome? Is it from quality of life? So the benefit of the GPC is in the fact that you can trace back from which outcome the overall result is coming from. And I also repeated the analysis for each of the treatment periods separately, and there again we confirmed that it's mostly the first treatment period that shows the treatment effect. Again, probably because of the carryover effect in period 2. So in fact, I hope I've been able to show you that general pairwise comparison methodology is quite flexible. You can combine any number of outcomes, including the patient-relevant outcomes, such as quality of life. It does account for correlation between these outcomes, and it may, as shown in the EB trial, it can increase your power. That depends, of course, if the treatment effect is on both outcomes compared to univariate outcome. And on top of that, we also have quite easy interpretable treatment effects, and it can give you insight on how it comes to the overall results. Now, practically, we have the exact permutation test, which is easy, fast, and very precise in very small samples. And there is software available in SAS, in R, and there's also, and I think Mark may discuss a bit further on that, there's also a program in development in Python that allows you to use this methodology quite in a straightforward way.